Okay, can you guys hear me?
I can hear you.
Well,
Yes.
thank you. You'll notice this is a new computer and it's not displaying correctly. In fact, my screen is totally black, and I don't know why that is. So you'll have to live with that. In a moment, I'll show my screen, in which case you'll see what is on my screen. That you can see. Any questions? I'm not quite ready. I just got this computer today. Now I'm looking for my syllabus. I'm not finding. Let's try fall 2022. There it is. Yep. Sign in to set up Office. I thought I already had Office. Unlicensed product. I'll have to go into Clark College to get Microsoft Word and PowerPoint to work. I was just there and it worked there, so I thought it was already logged in. All right. Let me share my screen. So you should be able to see my screen now. We're a little behind schedule. Don't remember what chapter we're on. Chapter 5. So today is October 27th, and we're supposed to be starting chapter 6, but we're behind schedule. Any questions about any of that? We will be having lab 7 in the lab today at 6:30. I'm not using my headphones, so hopefully everything is okay. It's because I'm not really quite ready. Oh, look at this. This came up again. What slide were we on? I wrote that down because I can't put it in PowerPoint when I don't have PowerPoint totally functioning. Slide 51 is where we ended. All right, so I talked about the electron transport chain and how it happens in the mitochondria inside the inner matrix, inside the inner mitochondrial membrane, and the electron transport chain happens across the inner mitochondrial membrane. Now in prokaryotes, this happens in the cell membrane. So electrons are passed from one carrier molecule to the next in a series of redox reactions in the chain of proteins in the membrane. Electrons are passed through the chain. Some of the proteins pump hydrogen ions across the membrane. And how they do that is when the electron is going through the protein, they give the protein the energy it needs to pump hydrogen ions. Actually, the hydrogen ions are pumped against the concentration gradient. So it's working similar to an electron transport protein, except ATP is not being used. It's the energy of the electron. That creates a concentration gradient of hydrogen on this side, as well as an electrical gradient. There's more hydrogen plus charges on this side. And that creates the proton motive force. There's a twofold gradient. These hydrogen ions are going to move from this side of the membrane to that side of the membrane. And that's potential energy difference. The cell uses that potential energy difference to make ATP. The hydrogen ions can only move through this channel protein, and this protein is an ATP synthase enzyme. When the hydrogen ion moves through the channel protein, it gives this channel protein the energy it needs to phosphorylate ADP and make ATP. Any questions about that? How efficient is this in making ATP? About 40% of the energy from glucose can be made into ATP. And you might say, well, 40% isn't very good, isn't very efficient. However, about the best engine that we've ever made, meaning humans have made, has about 20% energy efficiency, converting the energy in gasoline into the energy of movement. And so the cell is about twice as efficient as the best engine that humans have ever made. Any question about any of that? All right, the making of ATP, that's phosphorylating ADP, and it's called oxidative phosphorylation because you use oxygen in aerobic respiration. However, this is just another name for the electron transport chain, so that's the term I'll use. Aerobic respiration, the counting of ATP, is in prokaryotes, we get 38 ATPs for the oxidation of glucose in aerobic respiration. Now for eukaryotes, it's between 36 and 38 ATPs. As I mentioned, the heart, the liver, and the the heart, the liver, and the kidneys are more efficient, and they can generate 38 ATPs per molecule of glucose. All other cells in the human body can only generate 36 ATP per molecule of glucose. The overall equation summarizing aerobic respiration is glucose, C6H12O6, plus 6 oxygen molecules, meaning 6 O2, and 38 ADP, and 38 inorganic phosphates, generate 6 CO2, 6 water molecules, and 38 ATP molecules. Now remember, the oxygen is not converted into CO2, it is converted into water. All of the CO2 come from the burning of glucose. Any question about any of that? So this is summarizing the aerobic respiration. We get 2 NADH from glycolysis and 2 ATP from the Krebs cycle and the the preparatory step. We get 8 NADH, 6 CO2, 2 FADH2, and 2 ATP per molecule of glucose. And then from the electron transport chain, we get 32 to 34 ATP. So the total in a eukaryote cell is 36 to 38 ATP per molecule of glucose. 6 oxygen are come into the system and then 6 water come out. Any question about any of that? All right, anaerobic respiration uses a different final electron acceptor than aerobic respiration. Oxygen is not used. It depends on what form of anaerobic respiration we're talking about for what the final electron acceptor is. So we're just going to say it's a molecule other than oxygen. It could be like nitrate, nitrite, sulfate, iron. Okay, it depends on what form of anaerobic respiration. Generally speaking, in anaerobic respiration, we do have glycolysis and the Krebs cycle. They do occur, and there is an electron transport chain, but the electron transport chain of anaerobic respiration is shorter than in aerobic respiration, and therefore the yield of ATP is less. It depends on which form of anaerobic respiration we're talking about, how much ATP we generate, but it will always be between 2 ATP and 36 ATP. Remember, prokaryotes in aerobic respiration generate 38 ATP. So the best anaerobic respiration will be 36 ATP. And I'm not sure of what the worst is. If we're talking about fermentation, and some people do call that anaerobic respiration. I'm not, because there is not an electron transport chain in fermentation. I think the worst would be 4 ATP, but fermentation generates 2 ATP. Therefore, anaerobes tend to grow more slowly than aerobes, and that's because they generate less ATP than aerobes. Any question about any of that? All right, so I briefly mentioned fermentation. This is showing you an overview of actually respiration in general. This isn't necessarily aerobic respiration here. Because oxygen is not coming. Oh, I guess it is right there. Oxygen's coming in. So that is aerobic respiration. And then fermentation. Fermentation does have glycolysis. It does not have the Krebs cycle and the electron transport chain. Instead, it has fermentation after glycolysis. In fermentation, we do have oxidation of glucose into, well, some molecule of the fermentation end product. Glycolysis occurs, creating pyruvic acid, and it is during glycolysis that fermentation generates ATP. The only generation we get is from glycolysis, so we get 2 ATP molecules per molecule of glucose. Any question about any of that? All right, so in fermentation reactions, we first have glycolysis happening, making the 2 ATP. We do generate 2 NADH, and then the cell, if it's engaging in fermentation, it proceeds with a fermentation reaction. It's usually 1 or 2 steps. I'm only going to talk about 2 fermentation reactions. Both of them have 1 or 2 steps. In fermentation, you take pyruvic acid and you make the fermentation end product. Come on, right here. And then you take the NADH, which is generated in glycolysis, and you convert that into NAD+. Now, why would a cell, like our muscle cells, can engage in both aerobic respiration and fermentation? Why would a cell convert NADH into NAD plus in fermentation, if it could keep the NADH and then eventually send it through aerobic respiration where we would get much more ATP. So why would the cell engage in fermentation and use up the NADH rather than store it?
It would do that if it wouldn't have enough access to oxygen, right?
Yeah, it does this if it doesn't have enough oxygen, and it has to use NADH in fermentation, and it recycles the NAD plus. If you have NAD plus, then the cell can engage in glycolysis and then engage in fermentation. So if the cell is short on oxygen, it'll switch to fermentation, and it won't store the NADH because it needs to have NAD plus to engage in glycolysis right there. And while the fermentation is occurring, we are making some ATP. Okay, so that's why the cell doesn't store the NADH and just wait for oxygen to become available because it wants the ATP immediately. So it uses up the NADH to engage in the fermentation reaction. Any questions about any of this? All right, so your muscle cell do engage in fermentation, but only when oxygen is limiting. Whenever oxygen is not limiting, the cell will engage in aerobic respiration, and then it can get much more ATP per molecule of glucose. However, if oxygen is limiting, the muscle cell will engage in fermentation to generate ATP, because as long as the muscle cell can generate ATP, the muscle cell can continue working. Any question about any of that? So when your muscles are really working, they're burning, they're actually creating lactic acid, and that creates the muscle burn. They are engaging in fermentation just to make ATP so the muscle cell can continue working. Once the glucose runs out, no more fermentation can occur. Of course, there's no
I'm
oxygen.
sorry to interrupt, but I can't hear anything. I don't know if anybody else is having that issue.
Can everyone else hear me?
Never mind, it's working again. Might be my Wi-Fi.
I had the same problem. I was about to say something in the chat, but I can hear you now, Dr.
Yeah,
Bolt.
did you see that? A message just showed up on my screen saying that the internet connection is unstable. So that's probably why you weren't hearing me. Anyways, your muscle cell, when there's no glucose, the muscle cell can engage in fermentation.
I couldn't hear anything.
There will be no ATP. And the muscles will stop working. And you might see this occasionally in the Olympic athletes. Usually during one Olympics, there will be one Olympic athlete who will be doing something like running, and then they just collapse. It's because they have no more glucose in their muscle. If it's the winter time, the coach runs out and puts the blanket over them, and their heart and the lungs are working like mad. That will bring both glucose to the muscle as well as oxygen. And then the Olympic athlete will walk up, stand up, and then walk across the finish line. Any question about any of that? All right, there are two types of fermentation we want to briefly discuss. Lactic acid fermentation, that just puts on one fermentation reaction after glycolysis, converting pyruvic acid into lactic acid. This is actually very common, lactic acid fermentation. It's the fermentation that makes sauerkraut, yogurt, most cheeses like cheddar cheese or I don't know, Munster cheese. This is the reaction that occurs when we're making bread, leavened bread, lactic acid fermentation. Although, excuse me, that's not bread. That isn't bread. Bread is alcoholic fermentation, which is the next one I want to talk about. It puts two fermentation reactions on. You don't need to know the first one. You make 2-acetaldehyde, and then you do another fermentation reaction making ethanol from pyruvic acid. Ethanol is a 2-carbon molecule. Pyruvic acid is a 3-carbon molecule. So we do get some CO2 given off. And in wine, they do it in an open barrel or a casket. And so the CO2 just vents to the air. However, if you were to cap the bottle of wine, we don't call it wine, we call it champagne. And the CO2 creates the bubbly of champagne. And then beer, you do create it in a closed environment. And so that creates the fizz in the beer. There are other fermentation reactions. I'm not going to talk much about them. There's propionic acid fermentation, and that's how we make Swiss cheese. So Swiss cheese is an exception. There might be another one that's an exception too. But there are different fermentation reactions, and industrialists use these for making different products like acetone, formic acid, acetic acid. And of course, like I said, lactic acid, that would be what we use to make yogurt, and ethanol, alcoholic fermentation. That's what we do to make ethanol that they put in your gasoline, as well as this is the same reaction for making leavened bread and wine and beer. Any questions about fermentation? This slide is just showing you the different fermentation end products. You only need to know ethanol and lactic acid. And then the industrial commercial use, the starting material, which you certainly don't need to know. And you don't need to know the microorganism that does it either. I will say there's many bacteria that engage in lactic acid fermentation, and they're showing you some here. There's really, for ethanol, it's mainly just yeast, Saccharomyces cerevisiae, which engages in ethanolic fermentation or alcoholic fermentation. And then here's a slide showing you a summary of aerobic respiration, anaerobic respiration, and then fermentation. The growth conditions, aerobes have to have oxygen around. Anaerobes do not use oxygen. So oxygen does not need to be around. And then fermentation can be either in aerobic conditions or anaerobic conditions. Generally, anaerobic organisms like to grow in anaerobic conditions, but there are exceptions, okay, where the organism can engage in fermentation under aerobic conditions. And one is yeast making alcohol. And I think also another one would be some bacteria that make yogurt can engage in that's fermentation, never mind.
Can
I don't know
anyone
if it's happening
hear?
for anybody else, but I can't hear anything either.
This
Yeah, I can't
slide
hear
is
anything
just
either right now.
This slide is just showing you, let me see if I can shut anything down. I don't think I have that much open. Oh, come on. Hopefully this won't shut us down. Your connection is unstable. Nope, don't want to do that. I can shut that down. Shut that down. And I can shut that down, although I'll have to go there before I start the lab. I have to have that on. Let's go ahead and shut this down too. Now that shouldn't interfere with the internet. Let me go ahead and shut that down too. Well, no, I want to get on chapter 6, so let's not shut that down. All right, hopefully that'll help a little bit. I'm not sure. Why do I have 2 of these? Hopefully this will be better. Let's talk briefly about lipid and protein catabolism. Glucose is primarily the agent used in respiration, both anaerobic and aerobic, as well as in fermentation. However, it doesn't have to be that way. Any carbohydrate can be used. Almost all carbohydrates are simply broken down and then converted to glucose.
I can't see anything. Can anyone see anything?
I also can't see anything right now.
Can you see it now?
Oh,
Yes.
All right. I'm not sure why the screen my screen thing discontinued. I didn't shut that down. Anyways, my point is that carbohydrates can be used. Lipids and proteins can be used as well. So almost any food source can be used by energy to make ATP. There is a problem, and that is large molecules cannot cross the cell membrane. So a lipid and a protein are large molecules. They cannot cross the cell membrane. Doesn't matter if this is a membrane of a bacteria or the membrane in your gut cell. Lipids and proteins cannot cross the cell membrane. They're just too large. So what the cell does is it digests the lipids and the proteins as well as large carbohydrates outside the cell. We call that extracellular metabolism, where they secrete enzymes outside the cell to digest the lipid and the protein. And then they're made into smaller molecules. Lipids are made into fatty acids and glycerol, and protein are broken down into amino acids. And then these smaller molecules are brought across the cell membrane with a transport protein, meaning they can't move across the membrane, but they can with the transport protein. And so the cell produces lipases and proteases to digest the lipids and proteins outside the cell. I think I'll just skip this and move on. The relationship for how the lipids and proteins, as well as carbohydrates, are digested are shown in this slide. Glucose, of course, right here. Almost all sugars and carbohydrates are converted into glucose sent through aerobic respiration the normal way, or if we're talking about anaerobic respiration, in the normal way anaerobes are. Lipids are a little different. They'll be broken down into glycerol and fatty acids. Glycerol is then converted and joins the Krebs cycle. Not the Krebs cycle, sorry, the glycolysis. And it doesn't start at the beginning, it starts in the middle. You don't need to know exactly what molecule, but glycerol does enter aerobic respiration, and the in the, I just said it, glycolysis. Fatty acids are broken down into 2 carbon chunks, and then those 2 carbon chunks are converted into acetyl coenzyme A, and then they join the Krebs cycle as acetyl coenzyme A. Proteins, it depends on what the R group is. The amino acid has to be pulled off, and then some of the R groups will be converted into pyruvic acid, and really that's the end of glycolysis. Some R groups will be converted into acetyl coenzyme A, meaning they're cut into 2 carbon groups, and then they join the Krebs cycle. Other amino acids join the Krebs cycle near the end of the Krebs cycle. The point is, all the molecules eventually get converted into ATP, meaning they'll be burnt up and join the aerobic respiration. I've mentioned that aerobic respiration is the most efficient for converting the energy in glucose to ATP. ATP is going to be the energy source for most cellular processes, including active transport of molecules across the cell membrane, the movement of the cell, and the synthesis of new cellular components, which we call anabolism. Most cellular ATP is used for anabolism, like the biosynthesis of protein or DNA. All right, I've talked about anabolic and catabolic pathways as being distinct and separate. In reality, they're not. They are joined by common intermediates, and we call these amphibotic pathways. That's a metabolic pathway that can function in both directions. So they bridge the anabolic and catabolic pathways. This is showing you most of the bridge, meaning where the reactions can go both ways. You'll note that generally when we eat lipids and proteins, that they are broken down and then join the aerobic respiration. However, they're not used directly by most of us, like couch potatoes, like most of you right now. Only when you're very active would you directly burn lipids and proteins. And generally, us couch potatoes, which aren't doing very much, will break down the molecules like amino acid, and then they will build that molecule up into glucose. And then when they want to get energy, they will use glucose. Okay, so when you eat lipids, for example, it will actually be broken down and then converted into glucose by most of us. Okay. It is true a marathon runner can digest lipids directly in aerobic respiration. However, the marathon runner doesn't do that until they're 45 minutes in their run. So for the first 45 minutes, the marathon runner is using up glucose and carbohydrates and converting lipids into glucose. And then after 45 minutes of running, the glucose is all used up, and the marathon runner will still start digesting lipids directly in aerobic respiration. So glucose really is the preferred starting agent for aerobic respiration. Any question about any of that? And as you can see, humans are not very good at amphibolic pathways, making other molecules from another molecule, but we are pretty good about converting food into glucose. And this isn't showing you two pathways, because this is not a direct pathway, but it does, this can go here and then go up. So we do make glucose. And this is showing you also there, you know that if you eat too much glucose or too much even protein, it'll be converted into lipids. And what you do is if you're eating a bunch of protein, it'll be converted into glycerol and then fatty acids, and then they'll be making lipids, and then we store the lipid down as fat. Green plants are much better at amphibolic pathways. Many of their reactions can go both ways. Like I said, humans are really terrible at amphibolic pathways, but we're pretty good at converting one food molecule to glucose, or the other way around, glucose into fat, for example. Any question about any of that? All right. Let's briefly talk about photosynthesis. Photosynthesis is the conversion of light energy into chemical energy, which we call carbon fixation, where CO2 and water is converted into glucose. Photosynthesis is performed by cyanobacteria, algae, and green plants. And it makes an organic compound like glucose from simpler molecules like CO2 and water, so that photosynthesis is needed for all organisms, like animals get their food from plants. And even if you eat a cow, the cow is getting its energy from eating plants. Okay? What you might not know is that photosynthesis has two forms. There is cyclic photosynthesis and non-cyclic photosynthesis. You're probably familiar with non-cyclic photosynthesis, where in the construction of oxygen, you make 6 CO2, 12 water in the presence of light, creating glucose, C6H12O6, plus 6 oxygen molecules and 6 water molecules. Sometimes this will be simplified. You remove 6 water molecules here and here. I prefer showing the full one because all of these 12 oxygen atoms come from molecules of water. None of them come from carbon dioxide. The carbon dioxide is all converted into glucose. You don't need to know there is an anaerobic form of photosynthesis. It doesn't generate oxygen, it actually generates sulfur. What you need to know from this lesson is, like I said, there are 2 types of photosynthesis, one creating glucose, and then cyclic photosynthesis, which creates ATP. All photosynthetic organisms that I know of can perform both non-cyclic photosynthesis and cyclic photosynthesis. And which one they do depends on what they want to make. If they want to make glucose, they'll engage in non-cyclic photosynthesis. If they want to make ATP, they will engage in cyclic photosynthesis. Any question there? All right, let's talk a little bit more about cyclic photosynthesis, because you should have had non-cyclic photosynthesis in your Biology 160 class. In cyclic photosynthesis, the reaction starts with light, and the lights hit an electron in chloroform, and it excites the electron and then causes the electron to start moving. Let me see if I can blow this up. The electron moves through this protein in a membrane, and then this protein in a membrane, and then this protein in a membrane, and then comes back to this protein in the membrane. The electron actually comes back to where it started, meaning it, in this case, it came from chlorophyll. The electron will travel and then return to chlorophyll. Why it's called cyclic photosynthesis. The electron travels in a cycle. Any questions about any of that? As the electron moves through this protein, it gives the protein the energy it needs to pump hydrogen ions from this side of the membrane to that side of the membrane. These hydrogen ions are then used because they want to move to this side of the membrane, and they can only move through this channel protein, which is an ATP synthase. When the hydrogen ion moves through this protein, it gives the protein the energy it needs to phosphorylate ADP into ATP. And where have you seen this before? Nobody's going to take a guess? It's very similar to aerobic respiration. Cyclic photosynthesis, the generating of ATP. The difference is the light starts it, and then the electron is moving through a shorter chain. Any questions about photosynthesis? All right. Are you guys there?
Yeah.
Okay, good. This slide is showing you a summary of ATP production. The energy source can vary. It could be glucose. It could be food. For chemosynthetic organisms, it will be another molecule, a chemical molecule, but an inorganic chemical molecule. We didn't talk much about chemosynthesis. So the point is the electron source can vary. It can also come from chlorophyll when sunlight hits it. So the energy source is coming from sun. The electron carriers can vary. It could be NAD plus. We did talk about that. FAD can carry the electrons. And we didn't talk about it, but in photosynthesis, the electron carrier can also vary. This is similar to NAD plus, but it's a little different. And then the electron acceptors can vary. For aerobic respiration, the electron acceptor, the final electron acceptor is oxygen. For anaerobic respiration, the molecule is some other molecule other than oxygen. For fermentation, it's an organic chemical compound, a derivative of glucose. And in photosynthesis, it's not shown here, but the final electron acceptor in cyclic photosynthesis is actually chlorophyll, the molecule that the electron started from. Any questions about any of this? All right, if not, that's it for chapter 5. Let's move on to chapter 6. Microbial growth. Know the terms of this chapter and understand the rough outline. We're going to talk about the various requirements that cells have for growth. Understand that there are 4 phases of cellular growth, and compare the different methods we have for measuring microbial growth. When we're talking about microbial growth, we're talking about an increase in the number of cells, not an increase in the cell size. All cells have requirements for microbial growth. There are physical requirements that include things like temperature, pH, the presence or absence of oxygen. And then there are chemical sources required by living cells, such as a source of carbon, a source of nitrogen, sulfur, phosphorus, trace elements, and organic growth factors. Any question about any of this?
Can you repeat the physical part?
There's physical requirements that include the temperature the cell is at, the pH, the presence or absence of oxygen, the salinity. I guess oxygen would be a chemical one, sorry, because it's an element, not a physical thing. All right. Each species has requirements for temperature, where the species has the minimum temperature it can grow at, the maximum temperature it can grow at, and the optimum temperature it can grow at. Microorganisms are classified into groups based on their preferred temperature range. Let me blow this up. We have psychrophiles, and they grow best around 10 degrees Celsius. This is the temperature of your refrigerator. And generally, they don't grow much, if anything, above 20 degrees Celsius. And then they also don't grow much, if at all, below a minus 10 degrees Celsius. The reason why they don't grow below that is that they freeze, and cells can't grow when they're frozen. And they can grow at below 0 degrees because they create a antifreeze. But the antifreeze only can work down to about a minus 10 degrees Celsius. There are psychotrophs, and there are organisms that grow best at room temperature. There are mesophiles, and these are organisms that grow best around 37 degrees Celsius. All human pathogens are mesophiles because they grow best at our body temperature. Any question about any of that?
I have
And-
a question.
Yeah.
If it goes outside of the temperature range, does it start to die or does it just inhibit the growth?
It depends on the cell. Like our cells, if you put it at a temperature which we could not grow at, our cells would first get sick, the person would get sick, and then they would die with their cells dying. However, bacteria, it depends on the cell. If the cell makes an endospore, it would continue living, okay? But things like E. coli, if you grow them way away from, or you put them in an environment way away from their optimal temperature, they will eventually die. Now it is true that, well, let's say you heat them up, they'll eventually die because they can't survive in this heated temperature. When you freeze them, some of them would survive, okay? Freezing bacteria at best kills only about 50% of the cells. And then if it's an endospore, you're not going to kill any. Okay.
Okay.
Thermophiles grow best around 60 degrees Celsius. That's quite hot. And then hypothermophiles, these weren't even known when I went and had microbiology. These cells grow best around 95 degrees Celsius. Remember, 100 degrees is the temperature of boiling water. These classifications are not hard and fast. There is some variation, like Streptococcus pyogenes does grow best at 37 degrees Celsius, and we call it a mesophile. But Micrococcus luteus grows on our skin, and its optimal temperature is actually 30 degrees. We still call that a mesophile, okay, even though the optimum is varying by at least 5 degrees. And the optimum range that the minimum will be and the maximum will be can also vary on the species. So it does vary for different species exactly what the graph will be, or the figure will be. Any question about any of that? So the temperature ranges are not rigidly defined. I think the hottest anything has been grown at is 123 degrees, or maybe it's 126 degrees, something like that. Let me briefly talk about food spoilage. Let's see. When you have food between about 51 or 52 degrees Celsius and 15 degrees Celsius, bacteria can grow rapidly, and it can produce toxins which would spoil the food. Even if it doesn't produce toxins and you have the wrong bacteria growing, it would spoil the food. An example of that would be like mold. And so food spoilage happens in this range. It can happen above that, up to about 62 degrees Celsius, and it can happen all the way down to about 0 degrees. In the refrigerator, as you know, food can spoil. What happens is the low temperature bacteria are growing. And so after a week of having food in the refrigerator, you should think about either eating it immediately or throwing it out, because the food can spoil in the refrigerator after a week. And I'm not talking about things that are preserved. I'm talking about general foods without preservatives. And by preservatives, I mean things like salt and sugar. Food spoilage happens simply when the food is left at too warm a temperature or is stored in too large of a batch and doesn't cool quickly enough in the refrigerator. Most bacteria grow best between pH about 6.5 and 7.5. But all organisms have a pH, optimal pH, where they grow best at. Some cultures, many microbes produce acidic environment if you grow them. So the microorganism itself will degrade the environment. Oops, didn't mean to move. We can use pH buffers and culture media to minimize the shift in pH. Acidophiles are bacteria that grow in acidic or environments low in pH. So most organisms grow between 6.5 and 7.5 is their optimal, but there are some which grow at very low pH, like a volcanic environment where the pH is between 0 and pH 3. And as you know, bacteria like Hylobacter, I'm losing the name of that, pylori, Helobacter pylori, which causes ulcers, can grow in the stomach, and the stomach pH is generally around pH 2. That would be an acidophile. Microbes do have a requirement on osmotic pressure. If the microbe doesn't have a cell wall and it's living in an environment where there's more water outside the cell than inside, water will move into the cell and eventually bursting the cell membrane. But the cell wall can protect the cell membrane by preventing the cell membrane from expanding any further than the cell wall. There are organisms that prefer growing at high osmotic pressures. Such as hypertonic environments. This would be the organisms growing in the Dead Sea, for example. They like to live and grow in a hypertonic environment. We can use high osmotic pressure to preserve food, such as molasses. Why molasses can stay at room temperature for, well, years. Another example is we put salt in pickles to help preserve it. We do salt meat and other things to help preserve it too. Now, pickles also have, besides salt, do have vinegar in it, which also helps preserve the pickles. But salted meat, molasses, and salted fish are examples of high osmotic pressure we use to help preserve foods. What happens under high osmotic pressure? The microorganism, which like it falls into molasses, the water will leave, the cytoplasm will shrink, so cell membrane shrinks, causing plasmolysis. Halophiles are organisms that require a high salt concentration to grow. There are two types. There are obligate halophiles. They have to have high salt in order to grow. And then there are facultative halophiles. They do not require high salt to grow, but they can tolerate. Most organisms do not grow at salt concentrations beyond 2%, but one facultative halophile is, sorry, I'm losing what it's called, Staphylococcus aureus. It doesn't have to grow in high salt conditions, but it can. And it can grow in up to 20% sodium chloride, which is what your skin is after you haven't showered for a while. There are organisms that prefer growing in a hypotonic solution with low osmotic pressure. One example would be Elodea, a algae that obviously grows in water. And it's a cell wall that prevents the cell from increasing the amount of water being brought in, increasing the size of the cell membrane. The cell membrane can only expand up to the cell wall. Once it gets to the cell wall, it cannot expand any further unless there is a hole in the cell wall. All organisms have a requirement of carbon. Typically, half of the cell is carbon. So if we dry cells, half of the weight will be carbon, meaning half of the non-water weight is carbon. Carbon is used in structural organic molecules like glucose. It's an energy source, so it's an ATP. Heterotrophs use organic carbon sources, but autotrophs use carbon dioxide dissolved in either the water or the air. Nitrogen is another chemical requirement. You probably don't need to know this, but nitrogen, 14% of the dry weight is actually nitrogen. Nitrogen is found in amino acids, in proteins, DNA, RNA, and ATP. Nitrogen can be obtained from the degradation or decomposition of proteins. But many bacteria, besides getting nitrogen from degrading proteins, can get it from other sources, such as ammonia or nitrate. A few bacteria can even use nitrogen from the atmosphere. And these we call nitrogen fixers, because they pull nitrogen out of the air and then make it available to the ecosystem. And how they make it available is the cell will eventually die, and then it will release nitrogen from the cell. Any question about any of that? All right, sulfur is another element that has a chemical requirement for living cells. Sulfur is used or found in amino acids like thiamine and biotin, and that's about it. But sulfur is also in DNA and RNA. Sulfur is mainly obtained from the decomposition of proteins, but many bacteria can use sulfur in other forms, such as sulfate or hydrogen sulfide gas. Any questions about any of that? All right, phosphorus, the last chemical agent we're going to talk about that all cells need. Phosphorus is found in DNA, RNA, and ATP. So phosphate is an important source of phosphorus. Trace elements, another chemical requirement of all living cells. This is an element, an inorganic atom that is required by microbes in small amounts, why we call it trace elements. Usually trace elements are picked up from their environment, from either the food we drink or the liquids we consume, and then it's similar for bacteria in the soil. Generally, the bacteria get its trace elements from the soil. The water will dissolve it out of the soil and then make it available to the bacteria. Trace elements can require an enzyme cofactor like iron or copper or zinc. And that's because the cell needs very few of these agents. All right, cells may have an organic growth factor, which is a requirement. Not all cells have an organic growth factor that is a requirement in order for the cell or the organism to live. Can anyone mention an organic growth factor for humans? What is a molecule, which is an organic compound obtained from the environment that humans get? Nobody has a guess? All right, vitamins, like vitamin C, is an organic growth factor. We must obtain it from our diet because we cannot make it. So vitamins, certain amino acids and fatty acids are organic growth factors as well. Any question about any of that? All living organisms have a requirement for oxygen. Why isn't that picture showing? All right, I'll have to show you this picture later because I don't have it on this computer. I don't think I can put a note in here either. Yeah, I don't have the ability to write on the slides, so. I even checked this to see if that was working at Clark when I got this computer, and it did work there, but it's not working here. So I'll need to take it back to Clark. Probably it needs to just connect to the Clark network, and then I'll get PowerPoint and Microsoft Office to work. All right. We'll come back to talking about, let me make a note of that. Come back to talking about oxygen. Where do I have that? We just had that. Because I looked at what slide we were on, but I'm not finding it now. Oh, here it is. No, that's another one, but that's a word. Yep, oxygen picture. All right, oxygen is in a sense a toxic substance for all organisms, including aerobes. There are 4 toxic forms of oxygen. The first is singlet oxygen, where oxygen is boosted to a higher energy state. It's extremely reactive and is actually present in phagocytic white blood cells. The singlet oxygen, the phagocytic white blood cell can release the singlet oxygen on something the white blood cell is attacking, like a bacteria, and then use the singlet oxygen to kill the bacteria. Another form of toxic oxygen is superoxide free radicals. A free radical is a molecule that has an extra electron to the molecule. Oop, didn't want it. Oh, that's right. This is where we want to be. So this would be O2 with an extra electron. That's what that is. That's not a 0. That's a box. And then it has a negative charge from that extra electron. The trouble with free radicals are they are unstable. And as this molecule bumps into another molecule, it will steal an electron from the other molecule, potentially destroying the molecule or disrupting the molecule, and possibly mutating the cell if it bumps into DNA. So this symbol is an unpaired electron, and it's only found in free radicals. Free radicals are highly unstable because they steal electrons from other molecules. How our cells neutralize free radicals is we have the enzyme superoxide dismutase, or SOD, and it takes superoxide free radicals, combines them with hydrogen ions to form hydrogen peroxide and oxygen. Any question about any of that? Now, hydrogen peroxide is still toxic to the cell. In fact, that is our next toxic form of oxygen. It's any peroxide anion, such as hydrogen peroxide, shown here. It's toxic. That's why we use it as an antiseptic, because it kills bacteria. To us, hydrogen peroxide is not terribly toxic, at least in small amounts, because we have the enzyme catalase, which breaks it down. Hydrogen peroxide, 2 molecules of it in the presence of catalase, breaks it down into water and oxygen. There is another enzyme which can convert hydrogen peroxide into water, and that's peroxidase. If the organism lives in the air and it is an anaerobe, it must have the enzyme catalase or peroxidase, or potentially both. Otherwise, it would kill the cell. And peroxide anions, meaning free radicals, are made in the metabolism of glucose. They're made at low numbers. In fact, they're a mistake in the chemical breakdown of glucose. But it's okay for our cells because our cells have the enzyme catalase. As long as the hydrogen peroxide isn't copious, like you're bathing in it, our cells can break it down with the enzyme catalase. And there are some bacteria that lack catalase and they have the enzyme peroxidase. And so they can still live in the presence of oxygen because when hydrogen peroxide is made, generally by accident, it can be broken down by peroxidase. Any question about any of that? We will talk more about catalase in the lab. There is a fourth toxic form of oxygen. The fourth and the last is a hydroxyl radical. Note that this is a different hydroxide ion, a different molecule than the hydroxide ion, which has 2 electrons, so it's not a free radical, and it's different from the free radical, which has 1 extra electron. I guess it's this one. So it is similar that it has a negative charge. But it has 2 electrons making the hydroxyl atom has 2 electrons making it. A free radical, which is a hydroxyl radical, has an electron. And this is different from hydroxide ion or from an alcohol. Hydroxyl radicals are made in aerobic respiration in very low amounts, trace amounts. They're also made by ionizing radiation and when hydrogen peroxide reacts with certain metal ions. How our cells deal with hydroxyl radical is we make antioxidants to remove the hydroxyl radicals, as well as other free radicals. And 2 antioxidants which remove hydroxyl radicals happen to be vitamin C and vitamin E. Any question about any of that? All right, I've already mentioned that microbes that use oxygen to produce ATP are more efficient than microbes which do not use oxygen. And that's because they produce more ATP. Microbes are classified on the basis of the effect of their growth. Oh, that's why I don't have that slide. I was trying to think I had a slide here, but it's actually right here, the one I was thinking of. Cells have different oxygen requirements, which is shown in this slide here. Let's talk about the obligate anaerobe. An anaerobe cannot grow in the presence of oxygen. When we make a thioglycolate tube from the autoclave, the thioglycolate media reacts with the oxygen and then chemically removes it. So once we take the tube out of the autoclave, there will be no oxygen in the thioglycolate. However, oxygen will move in from the media, and this will create an oxygen gradient in a thioglycolate tube, which is shown here. At the top, there will be lots of oxygen, around 20%. And in the bottom, there will be little, if any, it will be close to 0% oxygen. And that's because the oxygen is removed by thioglycolate media. For obligate anaerobes, they grow in the absence of oxygen. They will not grow in the presence of oxygen because oxygen will kill the cells. They lack the enzymes the cell needs to detoxify before oxygen toxins. Note that oxygen itself, O2, is not toxic. It's the derivatives of oxygen which are toxic. Any question about any of that? All right, so if we have a thioglycolate tube and we put in a bacteria, if the bacteria only grow at the bottom and there's less and less growth, the higher up we go in the tube, then this organism is an obligate anaerobe. It can only grow in the absence of oxygen. Come on, mouse. And as you go up the tube, there will be less and less growth. I should have one dot right here to show you that there's less here and then less there. Any question about any of that? All right, the next easy one is the obligate aerobes. They only grow where there's oxygen. And as you go down the tube, you get less oxygen. So as you go down the tube, you'll see less and less growth. In the bottom of the tube, you should have no growth. At the top of the tube, you should have highest growth because that's where there's most oxygen. Did I mention that oxygen moves into the thioglycolate tube, creating an oxygen gradient? At the bottom of the tube, there'll be no oxygen. At the top of the tube, there's full oxygen. And as you go down the tube, there's less and less oxygen. Facultative anaerobes are organisms which can perform aerobic respiration, but if oxygen is around, the organism can fall back on an anaerobic form of making ATP. And that could be anaerobic respiration, or it could be fermentation. In a facultative anaerobe, they grow best at the very top, shown here. And that's because they can grow everywhere in the tube, but they'll generate the most ATP where there is the most oxygen, at least on the planet Earth. And as you go away from that oxygen requirement, the organism will not perform as well, not grow as well. And so when we go down the thioglycolate tube, oxygen will decrease and we'll see less and less growth. At the bottom of the tube, we will get slight growth because this organism is a facultative anaerobe. It can grow when oxygen is present and it'll grow best here, but it can grow where oxygen is absent because it is a anaerobe. All right, there are also the aerotolerant anaerobes. These organisms can grow uniformly well in all positions in the tube. At the top of the tube, it grows well because although it is an anaerobe, it doesn't need oxygen. It does have the enzymes it needs to detoxify the oxygen toxins, the 4 oxygen toxins. And as you go down the tube, you'll see the same amount of growth all the way to the bottom because it grows equally well in the presence of oxygen and in the absence of oxygen. Any questions about that? There is one more I need to talk about, the microaerophiles. They are aerophiles, so they have to grow in the presence of oxygen, but a microaerophile doesn't like 20% or more oxygen. A microaerophile is an organism that grows best at oxygen reduced from 20%. Any question about any of that? So a microaerophile will grow someplace in the middle of the tube. And as you go away from the middle, you'll see less and less growth. So I should have a dot here and a dot there. So less and less growth, the further you go away from the optimum, which is the middle of the tube. Any questions about any of this slide? If not, let's talk a little bit about culture media. I think I've talked a little bit about it already, but there's different types. When we're talking about one, it's singular, and we say culture medium. If we're talking about two different types of media, it's plural, and we call it media. So medium, we're talking about one type of media, and then media is plural. We're talking about many different types of media, and so that's how you use the two words. Remember, a culture media is a liquid or solid solution engaging in the media, containing the nutrients required for microbial growth. Usually when you get culture media in the lab, it'll be sterile, and that means there's no living microbes in it. The inoculum is the microbes you introduce into the medium to get the microbe to grow. The culture is what we call the cells and the media when they're together. I could say growing together, but actually only the cells grow, the media doesn't grow. Any questions about any of that? When we're talking about media, you should realize in microbiology, we have a very large number. The book is about that thick, and a good size book, about that size, by that size, listing the different types of media we have available in microbiology. And generally, even us lab instructors do not use that book very often. Okay, the point is there's just a whole bunch of different media because a book that's that thick, and each page is one media. So a wide variety of media are available. The purpose of the general culture media is simply to support microbial growth. A medium must provide an energy source. It must provide a carbon source. And an easy way of doing both of those in one is simply providing glucose. It must provide a source of nitrogen and sulfur and phosphorus. And you could put protein into the media to do that. The media must supply trace elements if the trace element is required by the cell to grow. What would be an example? Well, human cells need vitamin C. Of course, that's not a trace element. We do need copper and magnesium. Okay, so that would be a trace element. And then there are organic growth factors which can be required, such as vitamin C. If you don't get enough vitamin C, you'll become sick, and then you'll eventually die. You do not necessarily have to have a microbe that requires organic growth factors, like Staphylococcus epidermidis is an organism which grows on the human skin, which does not have a organic growth factor because it makes its own vitamin C and whatever molecules it needs. Agar is a complex polysaccharide derived from seaweed, and we primarily use it as a solidifying agent for our culture media. Agars are almost always in plates, meaning if you have a plate, it's almost always going to have an auger in it. And they're also used in tubes. I think I've explained this before. We have two types of tubes, and I'm going to have a hard time getting this. See if I can edit this. Ah, come on. I'm not doing that. See if I can edit it directly. Actually, I just do this one. So if I do make a mess, this is an extra copy. We have two types of tubes. We have a deep, where the media is just in the tube flat across. And then we have slants, where the media is at an angle in the tube. And usually we have the media, a little combination of both, where there's a straight across and then there's a slant on top of it. Not drawn very well. So let's erase that. Well, that's good enough. This is called the deep. This is called the slant. Oh, nobody mentioned this is past the time. So are there any questions? To slide 22. Is there anyone here?
Are you still
Okay.
meeting at 6:30?
Yeah, I'll still meet at 6:30. Because people will be expecting that. All right, I'm going to end this and I'll see you at 6:30.